Okay, this is where I'm going to end it, and I'll start the next YouTube video from here. Okay, so welcome to DNA 101 Part 2. So I ended the last screencast by saying that, that mistakes can happen during the synthesis phase of the cell cycle, and that it is that some of these mistakes or mutations may be repaired. Many of them are, but some of them are not. And so how is it that these mutations can impact the functioning of the cell, or those cellular traits, that is the proteins they produce? And then of course those proteins that the cells produce impact our traits. Okay, so we know that synthesis, or DNA, the copying of DNA happens during the DNA synthesis phase. Okay, and then after those DNA copies are made, the cell starts to prepare itself for splitting into two cells, again by enlarging, producing extra organelles, different kinds of proteins, enzymes that are needed to undergo the division process, and then the cell divides. Okay, in this case we're saying by mitosis. Okay, and then the next phase of the cell cycle is this first gap phase, and this is when a lot, not all, but a lot of the traits that are in, or the sequences that are present in these, that are copied during synthesis, are then transcribed and translated into proteins. And so what happens during transcription and translation? So how are those words, those letters, words, and sentences then used to make different proteins for cells and for different groups of organisms? Okay, so in this G1 phase, what generally happens is the DNA that is present in the newly formed cells is then transcribed into messenger RNA. And then that messenger RNA then is translated into amino acids, which are linked together to create proteins, which are essentially traits in different groups of organisms, or contribute to producing traits in different groups of organisms. So after this is the newly formed cell, this cell is in the G1 phase of the cell cycle, and you have your DNA in the nucleus, and that DNA is transcribed into messenger RNA. Okay, and then that messenger RNA leaves the nucleus to the cytoplasm, and that messenger RNA is then translated into amino acids and proteins. Okay, so this is the process, the simplest representation of transcription and translation for cells. So any of the mutations that happen during the synthesis phase of the cell cycle before this new daughter cell was produced, okay, would be passed on to this daughter cell, and it is during transcription and translation that those mutations will potentially alter the types of proteins that are produced by this cell, therefore potentially changing the traits in the daughter cell. Okay, now remember that DNA is composed of letters, which are composed of words, which are sets of 3 letters, and that those words are then are important in producing sentences. That is, how do you, that sentence is, this is a protein. Okay, so what happens is that DNA is transcribed into messenger RNA. Okay, and then in groups of 3 nucleotides, that messenger RNA is then translated into amino acids and proteins. And each of those little words of the messenger RNA are used to, during translation, to produce an amino acid. And then each of these amino acids is linked together to create a protein. So phenylalanine plus plus proline plus histidine plus arginine might make up a particular protein. Leucine, serine, histidine, asparagine will probably make up a different protein, okay? So I think the simplest way to understand transcription and translation in its simplest form, again, this is a very, very simple explanation of transcription and translation, but it'll suffice for our explanation, our course, is to really, the best way to understand this is to actually do it, okay? So again, we're going to go back to the University of Utah, to a University of Utah website. So here is an exercise where you can practice transcribing and translating DNA. So click here to begin. Okay, so here's your DNA strand. It's being, the double-stranded DNA is being separated into single strands. Okay, so here is your template DNA. And what you need to do in this exercise is you essentially need to transcribe this piece of DNA into RNA. Okay, and remember, RNA is different because RNA is made up of A, U, C, and G. Okay, so A would normally in DNA be paired with T, right? But it's not in RNA. In RNA, it is paired with U. Okay, and then you can just kind of go along and transcribe this piece of DNA. All right, so here's our messenger RNA, right? So now we need to, the process of translation needs to happen. So we need to take three of these letters will essentially code for an amino acid, and then these amino acids are linked together to make proteins. So we need to start this this translation, and most genes, when you are translating a piece of RNA, most pieces start with a start codon. In many groups of organisms, that's methionine. Okay, so what you can do is you can grab that methionine, and AUG is the start codon. Okay, so there's your first amino acid. Now you have CUA. Okay, so CUA, and then you have GAC and GUG and UUC. Okay, and then UAG, and that's your stop codon. Okay, so now what you have is you have a series of amino acids that almost always begin with a start codon. In most cases, it's methionine, and that gene sequence ends with a stop codon. So now you have created a protein. Okay, so this segment of DNA here codes for one protein and is called a gene. Okay, so you can practice this transcription and translation. And again, we're always going to start from template DNA. So for those of you who had molecules and cells, we're not ever going to ask you to start with non-template DNA. We're always going to start with template DNA, just to kind of avoid confusion for those of you who know a lot already about this process. Okay, so now what's important to think about is that a point mutation in DNA after transcription to messenger RNA, and then after translation from messenger RNA to produce a protein, a simple mistake or a point mutation, that is, instead of that happens during synthesis. So when this DNA, so this is your normal, let's say this is your normal quote unquote wild type DNA. An error happens in DNA synthesis, okay, where instead of a G being grabbed and made a part of the copy of DNA, an A is, okay. Now, when this this wild type DNA is transcribed and translated, it produces a particular protein. In this case, this protein will cause mice to have dark coats, okay. The simple point mutation that happened during synthesis, when it is transcribed and translated, will lead to a different protein. In this case, the protein will code for mice that have lighter colored fur. Now, you can then see how it is by natural selection, depending upon the environment, how it is that having a dark coat versus a light coat might be beneficial or detrimental, and might lead to the persistence of a mutation in a population, or the elimination of a mutation in a population, if you go back to thinking about evolution by natural selection. Okay, so these simple mistakes that happen during the synthesis phase and that are passed on to daughter cells, that then that mistake is then transcribed and translated in that daughter cell to produce different proteins, can have very, very important impacts on the next generation if that mutation is passed on from parent to offspring. And there are a variety of ways in which that can be passed on, and we're going to talk about that when I see you guys during the next lecture. These impacts of point mutations can be pretty profound. So here, if you have, this is a sort of I like this table because it defines some of the different types of mutations that can happen during synthesis. Okay, because there is redundancy in the genetic code in the formation of amino acids, then sometimes a point mutation can really make no difference in the type of protein that's produced by an organism. So sometimes mutations happen and they ultimately have no impact on the amino acid produced and the protein produced. Okay, so very minimal impact on the organism. However, changes can happen that do change the amino acid produced, and then therefore the structure of that protein. And that can be positive, may not affect the organism at all, or it can be potentially detrimental. There can be things called nonsense mutations, where you have a stop codon inserted into a gene, which causes premature termination of translation. The polypeptide or the protein is then very short, and this usually leads to, it's usually a bad thing. Okay. Another thing that can happen, another type of mutation that can happen is a nucleotide can be inserted or deleted from the DNA sequence, which can ultimately, which is called a frame shifting mutation, and not only, it doesn't only affect one of the amino acids in the DNA sequence, one of the amino acids produced through translation of the protein, but all of the downstream amino acids. This can be, it is usually deleterious because it's such a profound change in the protein produced. It can be something that is a positive, but it is usually deleterious. It is usually these missense mutations that can potentially be beneficial for organisms down the road. So again, just to review, what is a gene? A gene is a length of DNA in the primary sequence that encodes for information for building a protein. In other words, it starts with a start codon, it ends with a stop codon of some sort, and that is generally a whole protein made up of individual amino acids. Each gene is made up of different numbers of nucleotides. So the immunoglobulin gene, for example, has 220 amino acids and 616 nucleotides, okay, because each amino acid is made up of 3 nucleotides. The insulin gene is much smaller, okay, with 51 amino acids and 153 nucleotides. So you do have genes that make up, that are of different size, and that have more or less profound impacts. Those proteins may have more or less profound impacts on the characteristics of an organism. These gene-carrying, genes are in most eukaryotes are linear pieces of DNA, and these linear pieces of DNA are chromosomes, and the chromosomes are defined as these sort of gene-carrying structures consisting of a molecule of DNA and histone proteins, which also assist with the coiling and uncoiling of DNA, which we'll go into a little bit more when we start to think about mitosis in more detail on Tuesday. Human chromosomes there are 46 linear chromosomes in the human genotype. Each non-sex cell or somatic cell contains 22 pairs of autosomes and 1 pair of sex chromosomes. And there are some really interesting things that can happen in organisms that have linear chromosomes, like most eukaryotes. And that is, not only can you have mutations that occur during synthesis that could be characterized as different types of point mutations, or which can result in silent missense, nonsense, or frame shift mutations. But you can actually have whole chromosomal level mutations. And I want to just introduce this idea, and then we'll come back to this a little later on. But you can get what are called inversions, translocations, or additions or losses of whole chromosomes. So you can get chromosomal level mutations. And here's an example of an inversion. Here's an example of a translocation. And we'll talk a little bit more about these when we come back, when we meet together on Tuesday. There's more information about mutations and their potential impacts on this website. You guys are going to be required to read this as a part of the assignment that's substituting for your quiz today. And when I see you guys on Tuesday, I'm going to link this discussion of mutations back to the cell cycle, and then we're going to talk about what happens during cell division, and the type of cell division we're going to focus on first is mitosis, and then we'll go into meiosis in more detail a little bit later on. So hopefully this is enough of a basis that we can get everybody caught up on the structure of DNA, transcription and translation. In the assignment, you'll be asked to practice transcription and translation and to review information about mutations. And if you have any questions or concerns about the assignment, please send me a message. I'll help you as much as I can via the internet. But take a careful look, especially for those of you who haven't had this in a while. Some of you are kind of bored probably right now because you've had this very recently. But for those of you who haven't had this in a while, review some of the resources that are available to you, such as the Freeman text, the websites that I've referred to, and then of course come see me if you need more help. All right, guys, so I'll see you on Tuesday. Don't forget to do your assignment, which is due before class on Tuesday. That will substitute, it's 10 points, and it will substitute for your quiz. And then we'll have a regular quiz the following Thursday. So more soon. Talk to you guys later. Bye.